Today our guest speaker is David Allen from NIST, and he'll be talking to us about some of the activities that go on there and some of the stuff he's currently working on, and also some maybe opportunities for students at NIST, some vehicles they have for that. Okay, let me just give you a 2-second background about Dave. He's been at NIST for over 20 years, so he definitely knows the ropes there. He currently works in the remote sensing group. He got his BA in chemistry, his MS in environmental biology, and a PhD in earth systems and GIS from George Mason. Current research interests include advancing hyperspectral imaging for crucial applications related to medicine, defense, and the environment. Let's see, some of the other stuff he's going to be talking about in his talk, but let me just conclude by saying that he's a member of SPIE, ASPRS, the Council for Optical Radiation Measurements, and American AGU. And also he was awarded the NIST Bronze Medal in 2011, the NASA Customer Service Excellent Award in 2002, and the Hood College Leah B. Allen Astronomy Award in '96.
Digging deep for those facts. That's great. Thank you, Emmett. When Emmett first invited me up, I promised myself I would not come up until after the snow is done, until snow season is done. And I pretty much accomplished that. The problem is, last week we had our own 8 to 12-inch snowfall, so it really didn't make any difference. But it's great to be here. I had worked and met a number of students that have gone to RIT, and including staff. So it's great to finally be here and see what the Institute looks like and the great facilities that are being run here. So this talk, I should lower your expectations before I start, and that is, this is not the type of talk where you walk away with a lot of new fascinating facts. The purpose of this talk is just to give you an idea of what's going on in my lab at NIST and a little bit about standards, and hopefully this will open up discussions for the future of possible collaborations, and we have opportunities for researchers and students. So keep that in mind as I talk about these topics. If you see something that interests you, feel free to contact me. I had to put up a pretty picture to start. I didn't want just a bunch of words, so I actually have what I'm going to talk about later in the talk, that is the NIST Forest project. So this is our headquarters, the picture in the right-hand corner in Gaithersburg, Maryland. So NIST, the National Institute of Standards and Technology. One important fact is we're not a regulatory agency. We're not like the EPA. We don't enforce any laws. We're simply around to establish standards, disseminate standards, and to do research. So we have a mission statement to promote U.S. innovation and industrial competitiveness by advancing measurement science. So it's really that measurement science and standards. What kind of science is there that doesn't depend on measurements? It's really all measurement driven. So we work closely with the international standards community and other government agencies. A lot of times we have guest researchers from other institutes internationally. More local to my group, my remote sensing group is in the physical measurement laboratory, we call it PML. And we also have a mission statement here. And I'm not going to read the whole thing, but we provide NIST traceable calibrations and disseminate standards. So you're going to hear a lot about that. Further, PML works continuously at the outermost frontiers of metrology, not meteorology, measurement science, not weather. Devising tools and techniques for the ever-changing demands of science, new technologies. I mentioned hyperspectral imaging in the title. And that's key. This is a relatively new, how you define new technologies or not. Hyperspectral imaging is kind of a newer technology, and there's a need for standards to support that industry, that field. A little bit about standards. Standards is not something that you'll find young kids saying, I want to grow up to develop standards. They want to grow up to be astronauts, NBA players, rock stars, or whatever, but not to develop standards. It's not a sexy field. It's a field that kind of takes the right type of person that feels like they could make a difference or would like to see a field or industry move forward. But they're everywhere. We really couldn't exist as a modern society without standards. So what is the standard? There are a lot of different definitions. And this one gets us part of the way there. It starts off by document. And a lot of standards are documentary standards. But you could also replace that with a physical standard. In a lot of cases, we have optical standards in our lab that we develop and disseminate. The important point here is that standards should be based on consolidated results of science, technology, and experience. And it's typically driven by consensus, at least in the US. It's not mandated. So at NIST, when we talk about standards, a lot of the standards they revolve around the SI units, the International Standard Set of Units. So we realize we develop scales, we develop absolute methods for these scales. And any measurement that you have, any measurement scale, especially if it's traceable to the SI, has to have uncertainties. So if you don't have uncertainties, something's missing from the picture. It's questionable. So when somebody says something is traceable to NIST, an SI unit is traceable to NIST, it must have an uncertainty statement. Those things go together. So for more details on that, anybody that wants to think about standards and traceability, we have kind of an extensive explanation at this link, and I can make that available later. This and any other of the links. Getting closer to remote sensing now from standards. This is kind of a sampling in my laboratory of the types of optical standards that exist. So we have a facility to measure reflectance, BRDF, and specular reflectance. This is what was referred to as the STAR facility on the left-hand side. Let's see, does this work as a pointer? No. So the blue color tile is where the sample might go when we measure the reflectance of the sample. So if you have a calibration target and you're seeing a Spectralon sample, for instance, a commercial name, that scale is derived in this facility. If you're making measurements outdoors with a Spectralon plaque or some other artifact, we develop a number of these standard materials that we call standard reference materials. And we have a whole catalog of thousands of these different materials. Additionally, the radiance scale. So every remote sensing instrument is measuring, if it's for passive remote sensing using the sun as the source, there's some radiance response. How do you get that response? We establish that scale and transfer that scale. So there are a number of these. What's missing here is some standard that's called hyperspectral imaging standard. Those don't exist. What we need to do is think which are applicable, and we're in that process now. It's not well defined. There's this new and growing field of using and deploying hyperspectral imagers, but the best practices for reapplying the existing standards or meeting the demand for new standards is kind of the current area that I'm involved in. One of the problems that you run into with hyperspectral imagers is they have the ground-based systems, these compact imaging spectrometers. Awesome, thank you. All right. So this is an example of a hyperspectral imager made by Headwall. This is a veneer square, visible infrared through the shortwave infrared. And they typically have a wider field of view compared to what a satellite sensor or small radiometer might have, which typically might be a couple degrees. These have a field of view on the order of 30 degrees, full field of view. So what that means is at some distance they need to fill the aperture, and if you use an integrating sphere, perfectly good alternative, you need a fairly large one. Otherwise you're smack up against the integrating sphere, which could cause feedback and loading issues. So an alternative, and there are pros and cons of each, is using this method where you use a plaque, and this is on the order of 14 inches, and a lamp, an FEL lamp. This is a 1,000-watt lamp, and this is a challenge to get a picture of this because typically it saturates and you don't see anything in the background. So this is kind of a good photographic accomplishment to be able to get that dynamic range. But it's a fairly simple setup. At 45 degrees, 0 degrees from the lamp to the plaque, and 45 degrees to some sensor under test, where we could have a known radiance source, this combination, and determine the radiance responsivity of the instrument. It sounds simple enough until you start considering all of the aspects to it. So very simply, what it depends on is a lamp as an irradiance source hitting a diffuser, so reflecting off of this, and this distance is critical. So this distance is typically set at 50 centimeters, but measuring it, the sensitivity is on the order of each millimeter can throw you off by about 0.3% in total. So it's very sensitive to distance. That is addressable as long as you know what you're measuring from and to, typically the center of the plaque to an estimate of where the center of the filament is. The problem is you don't always know where the center of the lamp filament is, so we use this block as a reference. But look, this has some spatial extent. We're assuming it's the center, but it's an extended source. And not only that, but that distance changes over this distance, and we're using an imager, so we want to image this full field of view. So there are all of these other considerations in reality. There's the lamp irradiance itself, we measure that. Lamp intensity versus direction, the RDF of the plaque itself. Polarization effects, kind of a second order effect, but they're there. Spectral aspects, are they uniform across the plaque uniformity position, and so on. So we're studying this in order to help facilitate the use of this kind of tried and true practice for hyperspectral imagers or small compact imagers. So what are we considering? The lamp properties are pretty well known. BRDF can be well known in our lab, and comparison to an integrating sphere. So we have this information, and with that information we could take that and put it into a model and look at correcting it. So Eric Shirley, one of our theoretical physicists, has taken this on with that lamp information. I'm just giving a snippet of the study. So for 3 different lamps, you could see how much the distribution varies, and if you don't have this information, you can have a significant difference. So notice at the center of this distribution, if we normalize it to 1, we could have a drop as much as 8 to 10% or more. So in terms of your final uncertainties, if you ignore this artifact, you could be off considerably. You're not going to be in the 1 to 2% potential. You could be off by 10%. Okay. So with that model, with all of these factors taken into account, the lamp, the plaque, the distance, to some extent the imager, we take the lamp plaque system and use an integrating sphere as our presumed perfect distribution, and take the ratio The data points are actual measure values and the line is the model. So we have come up with a model that can explain this variability, but it's only for one line, it's not across the whole plaque. So what are those attributed to? Well, here's what the major components are. So starting with the least significant, the BRDF variability across the plaque or diffuser is probably the smallest contribution. And then the distance uncertainty, and that's not only the distance from the center of the filament to the plaque, but also as a function of displacement, X and Y displacement across the plaque. But the largest contributor is the FEL intensity, the variation as a function of angle. If we take all these into account, we can come up with an estimate of the total uncertainty. This is just to give you an idea of the thought process that needs to go into this. And I've been talking to Emmett and other people on the DEERSIG team about coming up with a DEERSIG model to be able to do this much more efficiently and do a sensitivity analysis with a lot of other variables in the scene. So I think it might be the perfect mesh of technologies in physics. Okay. On a very fundamental level, where does the optical scale come from for power? When we talk about irradiance of either of those lamps or radiance, the scale starts with this device. It's referred to as an absolute cryogenic radiometer. Cryogenic meaning very low thermal background with a black body cavity. So at the very bottom here, there's an entrance window and port and a cavity to absorb the radiation, which is characterized here. So what do? We insert a laser into this cavity and measure the increase in temperature in this low thermal background environment. And we close the shutter. Then we use resistive heaters to heat it up to the same temperature. And then we know what that power is, and there's an equivalency between the two, so we alternate. And we do this as a function of wavelength for the lasers and compare this to another instrument. So this is where the scale originates, an absolute cryogenic radiometer. So if you're traceable to radiance, this is likely where the scale came from. The scale in itself, none of these scales do any good if we can't disseminate them, if we can't share them with other entities like NASA, for instance, or other government agencies or end users, private industry. Using the lasers, we compare to the absolute cryogenic radiometer to a transfer standard, and this is basically a detector with silicon photodiodes that has a very high quantum efficiency. And wavelength by wavelength, we can compare scales. This facility is called spectral irradiance and radiance calibrations using uniform sources. They like clever names, so it's a circus facility. This has played a central role in transferring scales to the remote sensing community, but it also serves a whole other purpose, and that is laser lines are very narrow, near monochromatic, fairly high power, high intensity. When you're assessing spectral bands on a multispectral imager or maybe even a hyperspectral imager, you should only get a signal for that wavelength. You shouldn't get a signal in adjacent bands, but we do. There's no perfect instrument. So we use this to assess stray light levels in instruments. We can tune through and characterize the entire instrument, but what do you do with that information? You could say how bad it is. It turns out my colleague Eugene Zong has developed a correction matrix. So now instead of 1 × 10⁻³ level of stray light, 0.1%, you could reduce it an order of magnitude or two more to make it 10⁻⁵, basically just by making this measurement. This is great because a lot of optical designs are already set. Nobody's going to go back and redo the optical design, but we can improve the instrument based on characterization. Those are in the laboratory standards, and those are used for pre-launch characterization of instruments. By and large. But there are other needs. Satellite sensors, a lot of times when they go into space, their components degrade from outgassing or exposure to the space environment. So they need to routinely look at something that's well known. Well, the moon, it turns out, is a pretty good calibration target. It's fairly uniform if you're measuring in irradiance, and it doesn't change that much. It only changes to something like 1 times 10 to the minus 8th, a very small change. So it's extremely stable. The problem is it tends to go through phases and it's changing minute to minute. So in the past, the USGS had a program where they monitored the moon, if you remember, something called ROLO. This is taking it to the next level, where we're using spectrometers that were characterized at NIST, have extremely low uncertainties, coupled to a telescope in an area with fairly low atmosphere, like Mauna Loa, 14,000 feet or so, and observing the moon. So we're setting up an observation station in Mauna Loa to measure the moon over a number of years to come up with data so that any satellite sensor on orbit could look at the moon and use it as a SI traceable calibration source. And so how do we transfer that scale? Well, we have some lamp illuminated sphere at some other point, maybe across from our observation point. This is just some sample data for a given date of spectra. So this large spread, or the spectra that change. So if you're familiar with the solar spectrum, this is what it looks like, the irradiance coming off of the moon. That's above most of the atmosphere. Ideally, it'd be above almost all of the atmosphere if you're using it for orbit. So there's another program at NIST, and it's in collaboration with all these other institutes, and this is called AIR-LUCI, Lunar Spectral Irradiance. Again, using the moon, but in this case using a ER2 that flies above most of the atmosphere, is almost in space, which presents its own challenges because it's kind of in a harsh environment, extremely cold, almost in a vacuum, and it's designed, this package, to sit in this pod and track the moon. So this is something that's currently being worked on, and the trick here is to not only have a calibrated spectrograph, but also have an onboard calibrator that we can depend on when we do comparisons to the moon. So it's another onboard activity. I'm going to shift gears a little bit to an area that I'm heavily involved in, and that is hyperspectral imaging, not just from air and spaceborne, from the compact imager level. And the reason I'm kind of enthusiastic about this area is because it's an area that's growing in terms of applications. Air and spaceborne, typically Earth observing applications, and those are great. Those are rough estimates, just hundreds to thousands of applications. But as we go down to these lower cost sensors, more portable sensors, and this slide's out of date, don't assume that these are the cutting edge solutions, but just kind of a sample of what's available. As they come down in price and they're more portable, the number of applications grows by a couple orders of magnitude. And so this field's maturing, and there are people here that I look up to that have developed processing algorithms. The community, a big chunk of the community comes out of this institute for processing. I think as this field matures, when we know it's fully mature is when nobody calls it hyperspectral imaging anymore. It's just going to be something that runs in the background in a handheld portable device, and we don't think there's anything special about it. We just appreciate whatever information it gives us. I think that day is going to come in a decade or two. But we need standards to support us in this. So one of the big drivers in driving down the size and the cost of hyperspectral imagers, and this is kind of a transition period from these big government programs to small commercial products, are UAVs and newly evolving opportunities of small sats, putting kind of commercial level spectral imagers on small CubeSat platforms. Not to mention the use on benchtops, which I'm going to talk about. The problem is that when we see a hyperspectral data key, assume it's the same quality that we get from Avarice or Avarice NG or some other platform, and that's likely not going to be the case for these lower cost systems. So it's a balance between size and weight and power and making it compact, straight light, other issues, lower cost sensors. What can we expect? How do we make sense of the performance and ideally make them tied to the SI scale? So what we'd like is to have standards in place, standard methods, and Emmett's upcoming lab I see is leading in this area. Is it fit for purpose? Not everything needs 1% level uncertainties or even 5%. There's some applications that could do without that. The better question is, will it do what you want it to do? But you need to be able to determine that. I think there are a lot of people here, many people here are well versed in hyperspectral imaging, but for any folks that aren't, just very briefly, for red, green, blue imaging, we're using 3 spectral bands. In hyperspectral imaging, we're using hundreds of contiguous narrow spectral bands, sufficient enough that we could identify a spectral signature from the substance that's being measured. So kind of synthetic spectra, just to illustrate the point, from 2 spectral bands. If it were just a multispectral imager, we might be able to take ratios of these, but the full spectral signature can be presumed to provide somewhat of a fingerprint, depending on the material. But the point I'd like to make here is that when we think of spectral signatures, and we think of a material, we think for each material, there's an associated spectral signature. It's like a fingerprint, and it matches. And that's not exactly the case. There's a distribution. There's variability for different reasons. This is a small micro well dish, 5 millimeter in diameter with different substances. And when I measure these on the bench top, I get distributions. So it doesn't matter for most substances what the material is, there's some distribution. If it were one signature representing it, there would be 1 pixel in the scatter plot. So I'm looking at 2 spectral bands, spectral band A and B. There's always a distribution between the 2. This is just a collection of natural histograms or distributions from Earth remote sensing scenes. And in processing, the goal is to separate these different clusters. But there are distributions. Your classes are distributions of materials. Okay. So the way I look at this, and I think there are similar mindsets here, but just to paint the big picture, we start off with some sensor, a hyperspectral imager, and I'm using Avarice as the example, but ideally something more accessible. We have a scene, typically Earth remote sensing scenes, but now we're looking at other scenes, and it could be medical or an industrial process. There's some environment, and if we move indoors, maybe that's less of a factor. There's processing, and a lot of people that come to NIST and they learn about thermometry or other physical measurements, and there's some equation of state. And I tell people, this is a lot different. There are a lot of different methods for processing this data, which is good and bad. So people might ask, what is the best processing algorithm? And I would stop and pause and say, I'm not sure if that's the right question unless you know what your material of interest is. Because it's that spectral signature and that distribution that are really critical to be able to predict what your final data product is and what those uncertainties are. Remember, if we calibrate our sensor for radiance responsivity and we come up with a number of, say, 2% or 5%, what does that mean for the final data product? I would say that's not enough information without knowing what your material of interest is and what your processing algorithm is. So you need what's referred to as the image chain approach, or this whole workflow end-to-end analysis of some means. I got into hyperspectral imaging from our program on medical imaging. And so we had a program for about 5 years where we developed hyperspectral imagers and deployed them to the clinic, and this is looking at a pig kidney using one of the devices that we developed. And so it was very useful in looking at what was kind of a standard scene. The surgeon, in order to operate on the kidney and not have it bleed all over the place, about 20% of your blood gets pumped through the kidneys every minute. So they need to be clamped off, but you don't want them to be oxygen starved. So you can come up with a chemometric map of oxygenation or deoxygenation. That gives you a good illustration right off the bat. And you can see there's kind of this anomaly. In some cases, there's not just one artery to be clamped, there might be multiple. So it gives the surgeon good feedback into whether or not they've accomplished their task or if they've kept it clamped off too long. So this is, if you haven't seen oxy and deoxyhemoglobin spectra, they're really nice opportunities to see the changes as a function of concentration. On the benchtop, we developed a system with a spectral light engine to be able to assess this curve in our lab. So getting at what are the spectral signatures, what is the material of interest, we have to study what those spectral signatures are in order to go back and say, well, how good does the imager need to be? So I started a project where I worked with Ron Shu at Ohio State, and they had access to the hospital and surgeons and animal models. So in this case, this was routine to create what's called a skin flap wound in the animal and watch it heal. This was routine. What wasn't routine is to apply hyperspectral imaging and assess the degree of what's called ischemia, oxygen starvation in the tissue. So this is about 3 centimeters by 15 centimeters wide, and this is a Resnon, basically a push broom hyperspectral imager on a linear translation stage that we set up over this. So we did the same thing for burns, and you know, this is, you could talk about the medical ethics of doing these animal studies. What I would contend is, what we're trying to do is collect data so we don't have to do repeat studies like this every time we do a hyperspectral imaging study. Every time someone develops a new hyperspectral imager, they could avoid going to the clinic if we collect this data and have a way of using it in the lab to simulate what might actually occur. So this was very valuable data, and I'll mention it again in a minute and how that was actually used. Surprisingly, there are a lot of spectra out there that people just aren't that familiar with, and that includes things we look at every day, like our own human skin. So knowing the spectra of human skin is very valuable for a lot of different reasons. Medical imaging is one of them, but also there's a whole new industry of wearable devices, and there are security aspects and a lot of different aspects. What we found when we got involved in this is that there were not a lot of studies that provided sample spectra all the way from 250 nanometers to 2500 nanometers, let alone with uncertainties for those. And if you go to SPIE, Photonics West, BIOS, and you hear talks about people developing new devices and how they got great data, typically it's the researcher, N equals 1, they've examined themselves, they've demonstrated they can do it on themselves. The question is, before it's clinically accepted widely, can it be used on different skin types, different complexions? So we thought we'd do a study at NIST, and technically it was very straightforward. The IRB process was a nightmare. It took years to get approval just to measure the skin reflectance of 100 people, but we did it. And this is what it looks like. So this is the distribution, and this is available online. We published the method and the results. So it's been downloaded 500 times in less than 2 months. So I think people are using it. I've already been contacted by a couple people that are developing their own imager, and they like to show that, you know, they're within this range. So that's useful. Before this, if you did a survey of all the different studies, they were off by up to 100% from the mean. Okay, so the skin flap study that we did on the animal model how do we use this data? We have hyperspectral data cubes of the severity of the wound and the gradients, and we can pull out mean spectra for each of these different regions that were confirmed by a pathologist of the degree of severity. We have a device that we could take the hyperspectral data cube to and ingest it and reproject it to another imager. So we've taken reality, collected it, preserved it digitally, and now we could repeat it, recreate it in the laboratory so that the imager thinks it's in the clinical operating room again, looking at the pig subject. So just as a comparison, the original mean spectra then matched with the hyperspectral image projector, we call the HIP, you can see they're well within the variability. The variability isn't shown here, but they match very well. So we have no need to go back and do another one of those animal experiments. We have the hyperspectral data cubes that we call a digital tissue phantom. A little bit more about how the system works. So this was funded by the Defense Department to do simulations for imagers that were due to be deployed. And it covers both the V-near and the SWIR spectral regions. We use a supercontinuum laser, and that's projected into the system through some dispersion device. In this case, it uses a whole series of prisms, I think on the order of 20 prisms, and projects that onto a DMD, a digital micrometer device, which is about a million mirrors that have a binary state that could be selected or deselected. So basically, we could take our spectral signature on a pixel by pixel basis and match it to reality and reproject it into the imager under test. The nice thing about this is it's application agnostic. It can be used for any application, for medical imaging, security, environmental. So one of the great things at NIST is that we have a lot of resources, a lot of equipment. But what we don't have is somebody dedicated to this project. So I'm just going to plug the opportunity here for potential postdocs. Or it doesn't have to be the HIP project, but we have opportunities. We have a listing specifically for remote sensing that's somewhere in here. And this is in my group, so if you see a program or if you want to come visit NIST and talk about this, you're more than welcome to. This is a great opportunity, and they pay pretty well from what I understand. Okay. So determining the spectral signatures, this is another approach. So, and I think this is catching on. It's, I think a lot of people, since these compact imaging spectrometers are becoming widely available, a lot of people are looking at things. And I think this is a great opportunity to look at things that would typically be in an Earth remote sensing scene at a very fine spatial scale, because remember, most Earth observing scenes are mixed pixels. And if you're trying to identify a pure substance to some extent, it helps to go to a smaller spatial scale. So you can't go much smaller than a microscope on a bench top. So we saw value in this, and we've used a number of different instruments. This is kind of a basic push broom instrument with a improvement in a lens and a real microscope, a Nikon microscope. So we can get down to the micron or submicron level. Those are great, but they have their own shortcomings. So we built a hyperspectral microscope based on a tunable laser system. So this gives us extremely high power, extremely narrow spectral band, sub-nanometer spectral resolution. So really, we call it ultraspectral. We've looked at a number of different materials, minerals and backgrounds, asbestos, and a number of other things. And then mixtures. It's great to examine mixtures of materials. So this is kind of a schematic of that same tunable laser system. And just one of our first examples was looking at a granite slab, so we can get down to the grain level. Along the same vein, mixing different materials. So DeerSig, you can take a catalog of spectra and put them in a scene. In this case, you can take raw materials, throw them in a petri dish, and get kind of complex distributions of data points in n-dimensional space, and look to see how complex the scene is. And we've done comparisons from real substances you'd find outside into a petri dish, and it turns out it's as complex, if not more complex, based on various metrics we've used, like box counting techniques, fractal dimension. So it's a great way to generate data at the petri dish level, low level of material, to test algorithms. Again, another example where we came up with some very basic glass microbeads with and without rare earth oxides that have a very distinct spectral shape. And we made different concentrations of mixtures, and we'd expect some kind of non-linear mixtures or spectra. So another opportunity to examine that. Okay. Now I'm going to shift gears a little bit to kind of my bigger projects at the time. And most standards are things that are great opportunities to kind of simplify. They're very basic, easy to understand, or we can model them really well, but those don't represent reality or the complexities of reality. They aren't the actual thing that we're trying to measure in the environment. And that's what this project's about. So I'm trying to take advantage of the NIST campus, just like you have done at RIT. You've imaged RIT quite a bit. NIST campus is fairly extensive, and we have a kind of a nice forested area. So what is the problem I'm trying to address? And that is, we know in the carbon cycle, in the biosphere, that we're contributing a fair amount of CO2 to the atmosphere. And not all of that's being taken up. So there are biogenic forces, there are the oceans and terrestrial land biomasses that are taking up carbon, taking up CO2. But there's somewhat of an imbalance. So we can measure the fossil fuel emissions. We could do stoichiometry of how much fuel is coming in. We can measure it locally, and we can get the global mean from the Keeling curve. But if you notice, the Keeling curve has this annual, interannual oscillation, and it drops down. Why is it drawn down? Because the northern hemisphere has a lot more land mass, a lot more vegetation. The plants play a huge role in taking up the carbon, not only globally, but locally. So most of these emissions are typically around developed areas, the Northeast corridor from DC to Boston, but they're also vegetated areas. So if we try to estimate the amount of emissions, what does it mean without knowing how much is being taken up by plants? And that's the motivation for this work. In fact, for the North American Carbon Budget publication, there's a new one that's about to come out. It's expected to be published every 10 years. This is one of the biggest uncertainties. And the thing is, right now we have some number on how much plants are taking up. But how do we know that won't change under different environmental conditions? So what do plants need? They need sunlight, they need water, they need nutrients to some degree. But under different conditions, under different temperatures, CO2 concentrations, will they always take up the same amount? Will it increase or decrease? There's a big area of uncertainty there. So we're not sure what will happen in response to human activity. But potentially we can measure what's going on. So in our approach, we're focused on the Northeast corridor. We're here in Gaithersburg, just outside of DC. This is the NIST campus, and we have this large forested area. You say, well, it's not a pristine forest. It's not, it's surrounded by traffic and industrial processes, and that's great. This is an urban forest where the CO2 is being emitted and it's being taken up. So what we want to know is how quickly are the trees growing? How much CO2 are they taking up? And this is a remote sensing problem that goes back early Landsat days. Even NDVI are kind of basic metrics, but they're not tied that well to the biophysiology of the plant. But we think there's a potential for making Earth-observing measurements to estimate the productivity of the plants. So from the satellite view, airborne, and we would like to set up sensors to look at the trees from a distance. The thing is, for any optical standard, you need to characterize it. So this is very complex, but we're tackling this by characterizing the heck out of it using a lot of different sensors in the forest. So in the end, our data product, we would have net ecosystem productivity. That is how much carbon is being retained. And the initial metric is gross primary productivity, the kilograms of carbon per square meter per unit of time. So here's the NIST campus outlined in yellow. The forested area is on the order of 60 acres or so. And ideally, I might have a tower up in this area, but I didn't have that opportunity yet. I just got started last year. We have a building about 100 meters away. But this is large enough that we could use airborne and some spaceborne platforms for observation. Just kind of a look at another perspective, our observation point, and we set up a test plot of 1 hectare in this region, looking closer up at this. So this forest has been around for over 100 years, mixed hardwood, looks like this. In some ways it's not, you wouldn't call it a natural forest. It's missing something. It's missing any young trees in this area due to the heavy deer predation. That's not that uncommon in the Northeast corridor. We also have this invasive species, but it is what it is. We want to link the optical signature to this plot. So we did a survey with this team, and we surveyed every tree in this 1 hectare plot. We have all the species, the diameter for all the trees in here, and some of the down trees referred to as snag. There are a couple of really sizable trees that are on the order of 1.5 meters. So this is the observation platform. This, by the way, is the same building that is used to measure gravity, if you care about things like that. But they were using the 3rd floor, so I moved up to the 3rd floor, and I'm using what some people call the fire escape. I call it my observation platform, to look at this edge of the plot. So this is this building here with this field of view looking at this edge. So to instrument it, we set out instruments that give us the biophysiological aspects of this whole plot and strategically placed. So we have temperature, humidity, soil moisture, respiration of the soil and the trees. And most importantly, something called a sap flow meter, which is basically like measuring your pulse or your metabolic rate and temperature. So when the sap flows through the tree, the presumption is that that's used for photosynthesis. And you could see it when clouds pass over the trees, the photosynthesis slows down. For the optical measurements at this point, we're looking at something called CIF. I'll explain what that is in a minute. Phenocam, basically just what is the growing season when the leaves come out and when they drop. Thermal infrared, photosynthetically active radiation, how much available sunlight is there for growth, and kind of a non-observation point measurement of the CO2 concentration using non-dispersive infrared at this point. So at this point, we're just looking out the window or off the balcony from this point, but we hope to move up to the rooftop this year once we get the safety provisions or railing in place. One of the key measurements for this, for measuring the plant photosynthetic activity, is kind of a subset of hyperspectral imaging. Hyperspectral imaging can be used for just detection or classifying materials. This is kind of more to the extreme of measuring biological activity. And it's based on measuring plant fluorescence. So really simply put, chlorophyll dissolved in a solution, it could be in the leaf, in the chloroplast, will glow red. Why is that? Well, it turns out that the photons that aren't used in the electron transport chain in the thylakoid membrane in the photosynthetic apparatus either have two fates, either get converted to heat or fluorescence. Okay, so in the photosynthetic process, in other words, there's nearly a direct relationship between photosynthesis, the available photons, and the fluorescence intensity. People have been taking advantage of this from remote sensing platforms, and one of the newer opportunities is using OCO2, which was meant to sample the atmosphere, not for terrestrial plants, but it turns out it has narrow spectral bands, and you can come up with a map that looks like this. So this is gross primary productivity, and you can look at the intensity of the chlorophyll fluorescence. You can almost see the squares. These are very large spatial squares, spatial GSDs. The ESA has funded a mission, a dedicated satellite for this purpose. So this is seen as something that will measure primary productivity, not only for forest, but for agriculture and basically all the plants that are available. This is an opportunity to really refine our radiometric calibration practices. And there's some in this community that don't think this is going to be very plausible without good radiometric practices and standards. The reason is because this is your typical plant reflectance spectrum. And if you compare the reflectance to what you actually measure, you'll find you have a couple small peaks. And this is the fluorescence, this isn't reflectance. And the way you get that is through the oxygen A, if you're familiar with the atmospheric absorption features or the solar distribution. At 760 nanometers roughly, there's this absorption feature where you really shouldn't be getting much reflectance. So if you have fluorescence, it's backfilling this. And the total signal is only about 1 to 2% of the total signal. And imagine that if you have experience pulling ground reflectance signals out through the atmosphere. This is a very low level. If you have any instrument artifacts, stray light and so forth, poor signal to noise, large spectral bandwidth, it's likely this will not be possible to measure. So we're taking our laboratory calibrated instruments out to this test site and looking at individual trees that we've mapped out and measuring the solar-induced fluorescence for different species. And at this point, we're just kind of measuring day to day. What we'd like to have is really high temporal resolution, almost minute to minute, so we could see the activity. Again, this is the PhenoCam. This website's available if you want to pull up the website. We're on a network, so someone's developed a network of PhenoCams. But this is really the key. This is really what gives us our ground truth. So working in collaboration with Boston University and a student at Harvard has developed his own circuit for sap flow. So before there were a couple of commercial instruments, but not something that was battery powered that you could plug directly into the tree. So we drew a hole in the tree, put in a couple of resistive heaters and their temperature sensors, and as the sap flow increases, the heat gets dissipated faster. So you get a signal back based on the sap flow. So this is looking at some of the early data, day to day. And you could actually see clouds pass over. These are different depths. It turns out we're on maybe the 3rd or 4th generation now. We're going to deploy these in the next couple weeks before the growing season starts. So this team is in Florida testing them on avocado trees right now, trying to get them ready for the forest. Some of the other ground truthing is getting up in the trees and making measurements of respiration directly on the trees. And look at the species along that tree line. This is Lucy Hutira, Barry Logan, and Jared out there making measurements, including respiration. This is a custom respiration backpack that can be deployed. This is our CO2 signal. This is very valuable information as the trees collect CO2. They also go through respiration at night. So we have respiration from the trees, we also have CO2 emissions from the traffic going by. You could tell the difference between a weekday and a weekend from this signal. So notice that the global mean is around 405 parts per million. This is real data, corrected data, calibrated, and it will spike up to 600 parts per million locally where there's a lot of activity. In my lab, we're looking at the plants very close up. We've established this again, the medical imaging program, not only to measure the reflectance and absorption properties, but the scattering. So this is my guest researcher, Paul, who's developed the national scale for diffuse optical properties. We're taking advantage of that for the leaves. We think this is going to be really valuable because if we look at the absorption between species, they look very similar. If you look at the scattering, they're fairly distinct. So I think this is tulip and red maple and pin oak, sorry. So input data for deer sig. This is the type of thing that we would like to build a tree out of. Being at NIST, there are other resources, weather stations, and extensive instruments for measuring downwelling, spectral irradiance, shadow bands. The problem is that we're looking at the edge. Optically, we need to get up above the canopy, and it's a good thing we have nice collaborators. I think I understand they work with your group too. They're great to work with, Bruce Koch and Larry Corp, with a G-Light imager that has many of the same sensors. So they saw this as an opportunity to fly over the NIST campus, and these are the flight lines. So we collected data twice last year. This is the King Air 90 aircraft that they're set up in. And some of the early, they provide some really nice data products that give us the canopy view. So we look at the structure, the tree composition, and the hyperspectral data, very nice hyperspectral signatures. The problem here is that they're not going to be flying over very often. We appreciate it whenever they can. But we need to get up above the canopy on a regular basis, and that's where UAVs come into the story. So we need really high temporal and spatial resolution, and possibly different angles. A tower would only give us, it would never give us perfect nadir. We'd always be looking at some angle. So we think a UAV might do it. And then we'd like to add some of the same sensors that I saw in your UAV lab at kind of a combination platform. So I thought it would be good to get started on this before the growing season because there are a number of kind of tricks to the trade. And we're working with the University of Maryland that has a UAV program, and basically just RGB imagery, and then they have this MicaSense Red Edge sensor, multispectral imager, 5 spectral bands for plant reflectance. These are the flight lines. This is misleading. This is Google Earth, so it's during the growing season, but what we actually measured was during leaf-off conditions, and so there's a ground GPS unit. This is taking off from the test site. We have some of these trees instrumented here, but this is not the edge. The edge is back here. I'm not a camera person, so I would have to guess this is because it's a rolling shutter and not a global shutter. Somebody can correct me on that. I was happy to, after a couple tries launching from a site that was slightly lower, you need good overlap to be able to stitch together the mosaic of hundreds of images. So this is what it looks like with leaf-off conditions. We put some key points in here, white targets. But already, there's a lot of valuable information. What we need to do is characterize the scene really well, account for all the carbon, and that includes the leaf litter and the down trees. And I think we could probably develop products for that. So this is an opportunity for somebody that has the time to consider quantifying the leaf litter. Otherwise, teams go out and measure this manually, and it's very expensive. So I want to note that this project that I'm working on now is a whole team of people from Boston University, namely Lucy Hutier, who was, I think, on sabbatical at Harvard this last year, Bowdoin College, Harvard, a bunch of people from NIST and NASA, and probably some people I need to add to this list. So, in summary, this is responsible for a number of standards in remote sensing, and it's beyond just lamps, plaques, wavelength standards. And and it's an area of ongoing research, including newer technologies, hyperspectral imagers. So it's a current topic of interest. It's a little bit of a challenge. Can we take the old standards and just reapply them, or do we need new ones? And the hyperspectral image projector is one way we've addressed this. But one of the key takeaways here is we have a lot of opportunities for collaboration, and I think we've already kind of initiated some potential collaborations here. SURF stands for Summer Undergraduate Research Fellowship. I can give you the deadline if you email me. NRC postdoc program I had listed. And just guest researchers, if you have your own funding, you're welcome, and there's mutual interest, you're welcome to come to NIST and work as a guest researcher. So, and short courses. As Emmett can tell you, he's experienced one and he's coming back for another one. So I'll wrap it up there. I have no idea where I am in time.
Yeah, excellent. Thank you. So we're running a little long, but are there any questions, pressing questions? I'm sure everybody wants to get out in the hall. Yes, Dave.
How much of your time in the lab and all these projects is spent developing your own instrumentation, or is a lot of it just developing your own sort of methods and procedures to characterize existing instrumentation?
It varies dramatically depending on the project and the needs. There are times when, months were spent in the lab developing instruments, but it shifts. It's hard to say. I have a new emerging project for hyperspectral imaging of printed material, and I think we need new instruments to do that, new illumination sources. So I see myself going back in the lab for that.
Anything else? All right, let's thank our speaker again.